This is the Galvanic Cells Lab. And in this lab, we're going to use our knowledge of half reactions to make an actual voltaic or galvanic cell, which is the combination of two half reactions. Our materials include various metals. We have magnesium metal, along with magnesium nitrate solution. We have, I need to grab a piece of zinc. I'll grab that in a second. Zinc metal and zinc nitrate solution, lead metal and lead nitrate solution, copper metal and copper nitrate solution. We also have a porous cup, which can both hold a solution and a metal inside it, and allows for an ion exchange between the inside and the outside of the cup. This porous cup is made of the same material that a coffee mug would be made of, but it does not have the glaze on it, and that allows ions to flow between them. It won't let the solutions mix the two liquids, but it allows for the ions to flow. So it does the same job as a salt bridge would in a standard galvanic cell. And lastly, we have a beaker, which will hold another solution. We're going to measure our voltage using a voltmeter, and we have two wires that come out of it, a red, where the reduction half reaction will occur, and the black wire, where the oxidation half reaction will occur. Since we have four different metals, and we're going to pair them together, there are 6 different possible combinations. So if you follow the handout, there are 6 experiments to carry out. The first 3 all involve copper nitrate solution along with copper metal in the beaker. So I'm going to half fill this beaker with copper nitrate solution, and I'm going to insert a piece of neutral copper metal. Nested inside the beaker, we're going to pour some lead nitrate into the porous cup. Again, I'm going to about halfway fill this. And then lead metal will easily fit inside the glass beaker. Okay, so here's our galvanic cell. Copper and copper nitrate in the glass beaker, lead and lead nitrate solution in the porous cup that is nested inside the glass beaker. Now, one of the ways of clipping these wires together is going to produce a positive voltage. I'm going to try clipping at first the lead with the red wire and the copper with the black wire. And if I do that, and it may be hard to see from where you are, but our voltmeter is giving a negative voltage. That means that we've clipped the wires the wrong way around. So I'm going to unclip the red and the black and just switch them. This time I'll put the black wire onto the lead and the red wire onto the copper. And if you can see this, the voltmeter is giving us a positive voltage reading of about 0.6. So in my lab report, I'm going to record for experiment 1, the voltage is 0.6. And then it says which metal was reduced. It's the one attached to the red wire. Well, the copper metal is attached to the red wire. And which metal was oxidized? The lead, because that's the metal attached to the black wire. We've now completed experiment 1. Let's move on to experiment 2. I'm going to unclip at least one of these. I'm going to set the lead metal down. I'm going to pour the lead nitrate solution back into its beaker. And this time I'm going to, into the porous cup, pour some zinc nitrate solution. So zinc nitrate, I'm going to nest that inside the glass beaker. And now I'm going to use this opportunity to grab a piece of neutral zinc metal. And if I attach the black wire to the zinc, and I just left the red wire attached to the copper, and I'm noting on the voltmeter, do I get a positive reading or a negative reading? The needle moved up from 0, and it's at about 1.0. So we got the positive reading. This time we guessed right as to the order in which the metal should be clipped. 1.0 is our voltage. And the metal attached to the red wire is copper. So that was the metal oxidized or reduced rather. And the zinc is the metal attached to the black wire. So that's the metal oxidized. We're now done with experiment 2. Reading on to experiment 3, again, we have copper metal in copper nitrate solution in the beaker. And the zinc I'm going to switch out. So I'll just set the zinc down. I'll pour the zinc nitrate back into its storage container. And now we're going to try magnesium. So I'll pour a little bit of magnesium nitrate into the porous cup. I'll insert a piece of neutral magnesium metal into the cup. Gently lower it down. And I just left the red wire attached to the copper. We'll see if that's the right way to clip these wires. I'm going to attach a black wire to the magnesium, and I'm going to look for a positive voltage. I do not see one. The voltmeter is not giving me a positive voltage. So now I'm going to flip the wiring order. I'll unclip the black and the red wires. I'll put the red wire onto the magnesium. Make sure it pinches well. I'm still not getting something. Sometimes we get a little bit of gunk onto the wires. Let's try this red on copper one more time. There we go. We're getting a reading now. A pretty positive reading, if you can see from where we are. I'm getting 1.4 volts. And we got that reading when the red wire was attached to the copper and the black wire was attached to the magnesium. So that means copper was reduced, magnesium was oxidized. We've now completed experiment 3. We will keep for experiment 4 the magnesium metal in magnesium nitrate, but we're going to pour out the copper metal and copper nitrate solution. So getting ready for experiment 4, we're to put zinc, I'm reading the chart here, zinc nitrate solution in the glass beaker along with the zinc metal, and we'll put the porous cup inside. And I'm just taking a guess as to what order to clip the wires. Let's try putting the red wire to the zinc and the black wire to the magnesium. And good, I'm getting a positive reading here. I get, looks like about 0.75 volts. So for experiment 4, I'm recording a voltage of 0.75. The metal that was attached to the red wire in experiment 4 was the zinc. So note zinc as the metal reduced. The metal attached to the black wire was the magnesium. So write Mg for the metal that's oxidized. We've now completed experiment 4. We'll move on to experiment 5. This time, we're going to leave the zinc and zinc nitrate in the glass beaker. I'm going to pour the magnesium nitrate solution back into its container. And according to the setup for experiment 5, I should pour lead nitrate along with lead metal in the porous cup. So I'm inserting that. I'm going to nest those together. So lead and lead nitrate in the porous cup. I'm leaving the red wire attached to the zinc metal. And let's just see if we happen to get a positive voltage here. Looks like I'm not getting a positive voltage. In fact, it's dipping. The voltmeter is giving me a reading below 0. So that means the order in which the wires are clipped at present is not correct. I'll put the red wire attached to the lead, and then the black wire attached to the zinc. And now we're getting a positive voltage. It's quite small, about 0.3 volts, but at least it's positive. And the order in which these metals were clipped is lead was reduced because it's attached to the red wire, and zinc was oxidized because it's attached to the black wire. Last combination, experiment 6. We're going to leave the lead and lead nitrate in the porous cup, and we're going to pour out the zinc and zinc nitrate and swap it for magnesium and magnesium nitrate. So now in my glass beaker, we'll pour some magnesium nitrate solution and neutral magnesium metal. And then we'll very carefully put the lead and lead nitrate porous cup back into the beaker. I'm leaving the red wire attached to the lead for now, and just trying black wire to magnesium, and that, let's see, are we getting anything? Sometimes this one is a little funky. Yeah, there we go. And we are getting a positive voltage. Sometimes the alligator clip needs to have a nice contact point. I'm getting about 2 volts, 2.0 volts for this last experiment 6. And note the wire connections. Lead was connected to the red wire. Magnesium was connected to the black wire. So lead was reduced and magnesium was oxidized. Now that we've conducted the experiments and gathered the voltage data we need, we're going to do a little bit of calculating to compare the expected voltage to the voltage that we actually got. So we'll take a brief break and then go to the board. Now for the analysis questions. First, they ask us, why is it necessary to use a porous cup? Well, the porous cup was necessary to allow for charge rebalancing between the two half reactions. If you have a galvanic cell and you don't have a way for charge to rebalance between the oxidation half reaction and the reduction one, then the oxidation half reaction would just become extremely positive, and the reduction half reaction would become extremely negative because it's receiving negatively charged electrons. If you don't allow for a rebalancing of the charge after sending electrons down the wire, then the flow of electrons cannot be sustained. Okay, question 2 is probably the more interesting one. We need to think about, let's take each of the 6 experiments, and we have from our data table which metal was reduced and which metal was oxidized. It's the one that was connected to the red wire for the one that's reduced, and the one that was connected to the black wire for the one that was oxidized. Let's work through experiment 1 very carefully, and then once you've got the format, we'll go a little bit quicker for the other experiments. So referring back to the data table, in experiment 1, the copper was the metal attached to the red wire. So Cu, or copper, is the metal reduced. Now, in question 2, we're going to look at the table of standard reduction potentials and ask ourselves, what's the voltage of the half reaction that contains this metal? So I'm looking at question 2, and I'm scanning this list of half reactions for the one that contains copper. And as I go down, it's about the 5th one down. It says Cu2 plus 2 electrons yields neutral copper, and the voltage, a positive 0.34 volts. Okay, that's important to note. Now we're going to consider the other metal. So in experiment 1, the other metal, the one that was not attached to the red wire, was lead. That means it was attached to the black wire. I'm going to use PB, lead symbol. And then I'm going to look at the table of standard reduction potentials for the half reaction that contains lead. It's exactly 2 steps below the copper half reaction. And as written in the table of reduction half reactions, we have the electrons on the left side and a voltage of negative 0.13 volts. Now, it might be a little bothersome to see the electrons on the left side of the half reaction, since that's our metal that was oxidized. Keep in mind, they don't give us a table of standard oxidation potentials. They figure you can do the flipping of the voltages as you do your calculation. And that's just what's going to happen. So the next part of calculating this is to apply the formula cell potential is the reduction voltage minus the oxidation voltage. By taking the minus, you're doing the flipping of that half reaction so that you end up with the proper voltage in the end. So we're going to use the voltages exactly as they appear in the table of reduction potentials. So it was the 0.34 for copper. And we're going to subtract from that the negative 0.13 voltage for the lead. And you know from math class that if you subtract a negative, you're really adding that voltage. So it's 0.34 plus 0.13 gives us an expected voltage of 0.47. Let's follow that same process for each of the other experiments. So in experiment 2, if I'm looking at our data table, we had copper reduced. So once again, it's 0.34. And we're going to subtract from that the other metal in experiment 2, the one that was attached to the black wire, was zinc. So let's scan the table of standard reduction potentials for the voltage associated with zinc. It's about halfway down. I get a negative 0.76. The mechanics are going to be the same as the previous problem. So if you subtract a negative, it's the same as adding. 0.34 plus 0.76 gives us an expected voltage of 1.1. Experiment 3, again, we had copper as the metal attached to the red wire. So I'm going to put 0.34 volts. And from that, we'll subtract the voltage associated with the other metal, which is magnesium. So now I'm going to scan the table of standard reduction potentials for magnesium, and I'm looking at about the 7th one up from the bottom, towards the bottom here. And it's negative 2.37. So if effectively on your calculator, you're going to type 0.34 plus a positive 2.37, and that gives us 2.71 for our expected voltage for experiment 3. Continuing on, experiment 4, in this case, it was zinc is the metal that was attached to the red wire. So our first voltage in the formula is a -1. That's okay. And we're going to subtract from that the voltage for the metal that was oxidized. And in experiment 4, that's magnesium. So negative 2.37. Again, when you subtract a negative, you're making it positive. And this one works out to be a positive 1.61. In the end, a galvanic cell should always have a positive voltage. That's how we know that the reaction is spontaneous. There is such a thing as a non-spontaneous electrochemical cell, but we call that, it's not a galvanic cell, we call that an electrolytic cell. Anywho, moving on, experiment 5. Zinc, again, was the element attached to the red wire. And we know that its voltage was 0.76 or negative 0.76. And from that, we're going to subtract the lead voltage. Oh, no, I'm sorry. Look carefully. In the data table, it wasn't zinc attached to the red wire. It was lead attached to the red wire. So our lead reaction voltage was a negative 0.13. That one goes first in the formula. And then from that, we'll subtract a negative 0.76. So effectively, it's a negative 0.13 plus 0.76. And we get 0.63 volts for our expected voltage. Lastly, experiment 6. It was lead and magnesium. Lead was reduced, and magnesium was oxidized. So if we apply the formula, it's a negative 0.13 plus 2.37, and that equals 2.24 volts. Okay, so now we've got our expected voltages for all 6 experiments. Our observed voltages is what you wrote down in the data table. In our case, we had, just running down the list, 0.6, 1.0, 1.4, 0.75, 0.3, and 2.0. We can quantify the difference between what we expected to get and what we actually got through the percent error formula. You might remember this one. I'm going to give us some room here. I'll leave the expected voltages, but I'll erase everything else. We have some room to calculate. Okay, so our percent error can be found using an absolute value bracket. Always, it's observed minus accepted, or you could do accepted minus observed. It wouldn't make a difference mathematically. In absolute value brackets, all over the accepted value. Now, our accepted value is the one that we have remaining on the board here. It's what we had calculated from the table of standard reduction potentials. In experiment 1, our observed voltage was 0.6. Our accepted voltage is 0.47. So I'm going to take 0.47 minus 0.6 in absolute value brackets all over 0.47. And then 0.47 minus 0.6, you might say that's a negative 0.13. But remember, absolute value brackets mean make it positive. And then it would be time to pull out your calculator. I'll just type this in. And I get, rounding to the nearest percent, whole percent, I get 28% error. Don't worry, even if we get a really big percent error, that doesn't mean that the data is wrong, or it means we have a good deal of uncertainty in the result that we got. But that doesn't mean anything necessarily is wrong or bad. It just means we've got to be suspicious of that way of getting that number. Okay, for experiment 2, the observed voltage was 1.0, and the accepted is 1.1. You can subtract these in any order. It doesn't have to be accepted minus observed. It could also be observed minus accepted. So this time I went observed minus accepted, and we'll get the same thing. So 1 minus 1.1, you'd say that's negative 0.1, but the absolute value bracket makes it positive. So 0.1 over 1.1 gives us 9%. That's much lower percent error. Experiment 3. The accepted value is 2.71. The observed value that I read from the voltmeter was 1.4. Okay, so there's quite a difference here. And we'll times it by 100 to make it a percentage. Okay, so this time we'll probably get quite a large percent error, and that's okay. and I get to the nearest whole percentage, 48%. Experiment 4, I got an accepted value of, we got 1.61, and then our observed value is 0.75. This time I'll go accepted minus observed. And if we subtract that out, we get 0.86 over 1.61. And rounding to the nearest whole percentage, that gives us 53%. Okay, so students, as you can see, we're getting some pretty large percent errors, 48%, 53%. Maybe we'll get something even bigger. But again, it's not necessarily troublesome. It's just part of doing science, is getting unexpected results. Okay, experiment 5. We had 0.3 for the observed value, and 0.63 is our accepted. So I get 0.33 over 0.63. Remember, the absolute value bracket makes it positive. And we get, again, a pretty high percent error, 52%. Experiment 6, the accepted value is 2.24. The observed value is 2.0. So that gives us a 0.24 over 2.24. And I get 11% error. That's more like it, or a much smaller percent error. So we've got a range of percent errors. Even though the lab report doesn't ask us to necessarily think of sources of error, sometimes it's helpful to do that. We could think of lots of reasons why our numbers may be a little bit off from our expectations. Maybe the metal of the alligator clip, the little clippy metal wire, didn't attach very firmly to the metal that we were testing. That's one possibility. The metal we were testing might have had some gunk on it, just a little accumulated dirt, which affects the security of that connection. I did do my best to make concentrations of the solutions that were 0.1, but even so, using the best scales and best equipment that I've got, my technique could be an issue. So the solution was supposed to be 0.1 molar. Maybe it was a little off. Maybe it was 0.09 molar, or maybe it was 1.1 molar. That would affect the voltages that we observe as well. So all of these can play into the results. Thanks for your great work.